This title is a rather conceited title. And the excuse for that, my excuse for that, is that Ruth Karen insisted that I give this type of talk. So if you think it is conceited, it's not my fault. It's hers. But I, essentially, I'm going to review a number of things that have happened in my life, and my excuse is that all wisdom, as Ishla said, begins with memory. Of course, as one of your Baltimoreans said, the older I grow, the more I distrust the familiar doctrine that age brings wisdom. So, but I'll let you make that decision. At any rate, the first decade of my own life was featured by 3 vaccine-preventable diseases from which, in fact, I almost died. One was pertussis, severe pertussis. The second was pneumococcal pneumonia that put me into an oxygen tent for a while. The third was severe influenza, which actually did put me into coma, and I had Bell's palsy when I recovered. Now, in 1939, during that decade, the total advice of the Red Book with respect to vaccination was that every child be vaccinated against smallpox, immunized against diphtheria, against typhoid fever if necessary, and injected with rabies vaccine if the need arises. So in my lifetime, there has been a revolution in vaccines, and starting from very, very meager basis. So what have I learned during a lifetime? I'm going to give you 10 lessons that I've extracted from my own experience. The first is take advantage of every opportunity. Now, when I finished medical school and then went to the Epidemic Intelligence Service of CDC, at the end of my training, there was a list of assignments, and I insisted on going to the Wistar Institute Anthrax Investigations Unit. The Wistar Institute is a research institute on the grounds of the University of Pennsylvania in Philadelphia. I'll tell you in a few moments why I insisted. But anyway, I arrived at the Anthrax Investigations Unit, and soon after I arrived, knowing nothing about anthrax, having little interest in anthrax, there was a call from a pathologist in New Hampshire who said he thought he had a case of inhalation anthrax based on his autopsy findings. So I went up to New Hampshire and investigated the situation, and it turned out that there were 5 cases of inhalation anthrax in mill workers in New Hampshire. And as we learned, as we investigated and retrospectively realized that they had all had contact with a particular lot of goat hair coming from the Indo-Pakistan subcontinent, which was grossly contaminated with anthrax, the goats presumably having died of anthrax. Well, this graph shows the clinical course of the 5 patients, typical of inhalation anthrax, the prodromal period, and then the sudden septic collapse, toxemic collapse. 4 of the patients died. This paper, the paper we published in the late 50s, was actually republished after the bioterrorist incident in 2001, some 45 years later. So I think that may be a sort of a record. At any rate, at the same time, CDC was studying the protective antigen anthrax vaccine that had been developed at Fort Detrick. This is based on a protein produced by the anthrax bacillus, which forms a heptameric pore on the surface of cells of the host through which the lethal factor and edema factor toxins pass and, of course, kill the cells and eventually kill the host. So this vaccine, consisting of that antigen, was being tried in mill workers. And the end of the day, when we put together the results, the vaccine proved to be highly protective against cutaneous anthrax and probably against inhalation anthrax, although the number of cases was small. This vaccine is still I mean, it's been changed somewhat, but it is still the vaccine which is our protection against inhalation anthrax, aside from antibiotics. So the second lesson is find a mentor. Now, in medical school, I met this gentleman, Robert Austrian, who came from Hopkins to Downstate Medical Center, Kings County Hospital, a hospital distinguished for many things. For example, having been built without surgical operating rooms, but that's another story. Anyway, Bob Austrian was a wonderful mentor, both in the laboratory, but also on the wards. He was a diagnostician par excellence, and it was really a delight to see him do physical exams. But he also put bacterial polysaccharides on the map. That is to say, showing that invasive pneumococcal disease was still a problem despite antibiotics, and that a polysaccharide vaccine could protect, as he demonstrated in African gold miners. And the use of bacterial polysaccharides, as you know, began to generalize. After pneumococcal, it was Haemophilus influenza polysaccharide and meningococcal polysaccharide, both of which proved to be rather effective, in fact. But there was the problem of vaccinating infants, and the discovery by Oswald Avery back in the 1920s that conjugation of polysaccharides with proteins could improve the immunogenicity of polysaccharides was taken up by John Robbins and his associates, and so the first conjugate vaccines began to be produced. And as you all know, in the 1990s, these vaccines proved to be dramatically effective in controlling the three major pathogens, bacterial pathogens of childhood. My second mentor was this man, Hilary Kaprowski. Now, I told you that I fought to go to Philadelphia despite the advice of W.C. Fields, allegedly on his tombstone, rather here than in Philadelphia. But despite that advice, I fought to go there because I knew that Hilary Kaprowski had just moved to the Wistar. And Hilary was noted for his work, even then, on rabies and on polio, and I wanted to work with him. So I went into his office, basically, and asked him if I could. And he said, sure. So one experience that we had was to work on this virus, rabies. You can see the bullet-shaped particles of the rabies virus. At that point, in the early 70s, rabies vaccination consisted either of nerve tissue vaccine, 21 doses, or duck embryo vaccine, 21 doses, because they were poorly immunogenic. And of course, there were notable reactions, particularly to the nerve tissue vaccine, encephalitis, Guillain-Barré, and allergy to avian proteins were quite significant reactions. And I'll come back to this in a bit also, the rabies virus was adapted to growth in human diploid cell strains. Now, this picture shows Hilary and Tad Victor, who was a rabies virologist, and myself, and I was mock inoculating Hilary with the vaccine. In fact, we were the first three to take the vaccine, which was supposed to demonstrate our faith in the innocuity of the vaccine. This vaccine was essentially rabies virus grown in the human diploid cells and activated with beta-propiolactone, and it proved to be highly immunogenic. And this slide shows the antibody responses after intramuscular or intradermal inoculation, both of which are used today. The protective level is zero point 0.5 international units, and you can see that a 4-dose series gives protective antibodies. And indeed, it so happens in a way by coincidence, that this 4-dose regimen is now in this country the standard regimen for protection against rabies. In order to collect these data, one of the things I did was to visit just about every veterinary school throughout the country, enrolling veterinary students in these studies. Now, I can tell you that veterinary schools are located in some out-of-the-way places, and it was rather, I would say, depressing to be in a place where the only thing open at night was the Dunkin' Donuts. But anyway, we got through those studies, and eventually the vaccine was licensed and has become the gold standard vaccine against rabies. Lesson 3 is be careful what you do when you're young, because it may come back to haunt you. Now, as I said, I convinced Hillary to let me into the laboratory, and the laboratory that I was mainly working in was the polio laboratory. At that time, Hillary was developing attenuated polio viruses for oral administration. And so in the lab, we learned a number of things: selection of plaque mutants, neurovirulence testing in monkeys, the schedule for immunization, including newborn immunization, which was a new thing at that time, the effect of maternal antibody on immunization, the instability of attenuation in the gut, that is, the selection of reverted mutants, the identification of those mutants, the fact that virus was excreted for long periods of time and could spread to contacts, mass vaccination, I'll come back to that, and the problem of interference with OPV in the tropics. Well, Hillary's competitor at the time was Albert Sabin. And as you all know, Albert strains did eventually prove to be the better strains, or, well, one can argue about that, but anyway, his were the strains that were adopted for worldwide use and have resulted in the control of polio throughout the world. So that was, of course, a kind of a competition similar to the Gallo-Montagnier competition of recent years, and it was certainly at times a rather heated competition. But in the course of that work, I went to the Belgian Congo to do the first mass vaccination, and the reason that we went there was because Belgian scientists had come to Hillary saying that there was endemic and epidemic polio in the Congo, and could something be done about it. So the mass vaccination was done there and did show efficacy, although interestingly we showed that the efficacy was only about 60% in that region, which is still a problem, that is vaccination in tropical regions. But it was a successful campaign. However, in 1999, a British journalist published a book in which he proposed the following hypothesis: that AIDS did start, or was first noticed in the Belgian Congo, at least retrospectively, that the oral polio vaccine, the so-called chat strain, was studied in the Belgian Congo, that there was a chimpanzee camp associated with the polio work near what was then called Stanleyville, now Kisangani, and that the chimp simian immunodeficiency virus is genetically the closest virus related to HIV, so OPV must have been made in chimpanzee cells contaminated with SIV, which somehow then mutated to HIV. So that hypothesis received a lot of play in the newspapers and even in the scientific literature. So I spent about 6 months of my life, later in life, dealing with this controversy. And this is a picture of one of the items that I had retained in my files. I'm not going to read this, it's irrelevant. But the point is, I had fortunately kept a lot of the files that went back to the 1950s so that I was able to obtain information, retrospective information, which otherwise would have been lost. And that may be another lesson, never throw anything out. But the results of the investigations were that we determined, showed that no chimp cells had been in the production labs, that the chimps were used for neurovirulence testing, that PCR of the OPV lots used in the Congo were negative for chimp DNA, but positive, naturally, for macaque DNA, since the vaccine was made in rhesus monkey cells, that the same lots had been used in the US and Europe without, of course, producing AIDS, that there, even in the Congo, there was no geographical correlation with vaccination. And working with Beatrice Hahn and others, molecular evolution studies suggested that, in fact, HIV started in about 1931. That is, crossed over from chimps in the Cameroon, west of the Congo, not the Congo itself, because those are the chimps that carry SIV most closely related to HIV, acquired because of the practice of butchering and eating chimpanzees in the Cameroon and indeed in the Congo. Lesson 4 is the heresy of yesterday is the gold standard of today. And that relates to something that was happening at Wistar when I went back there after doing pediatric residency and opened my own laboratory. And that was that Leonard Hayflick at Wistar was working on cell strains developed from human fetuses. And what he showed was that those cells had limited lifetimes. That is to say, they would replicate for a certain number of passages, usually about 50, but then they would die, just as we all do. And so that these were not continuous cells, but they were, so to speak, pure cells. At that time, the first simian viruses were being discovered, and the fact that primary cells could be contaminated with other agents was becoming an important problem in vaccine production. So I took one of those cell strains called WI38 and applied them to rubella, because at that point in the early 60s, there was a huge rubella pandemic. As shown here, millions of cases, lots of complications in adults who had rubella, but also many infections in pregnant women, leading to thousands of fetuses dying in utero, and also at least 20,000 congenital rubella syndrome cases. It says 5,000 therapeutic abortions, but I can tell you from my experience doing diagnostic virology during the epidemic in Philadelphia that there must have been many more therapeutic abortions than that throughout the United States. We're able to calculate that 1% of all of the pregnancies in Philadelphia at that point were affected by rubella infection. At any rate, so we adapted, well, this is a rubella cataract, cataracts in an infant born after congenital rubella. So working in the lab, we learned how to grow virus in large quantity, for example, in baby hamster kidney cells. The diagnosis of maternal infection, isolation of virus from the fetus, the importance of placental infection to fetal infection, the interesting fact that the virus actually was distributed through all of the organs of the fetus. Of course, with others, many others, we described the outcome of fetal infection and worked on the mechanism of persistent fetal infection. That is, the virus not only infects, but the cells remain alive and the virus continues to be produced. And what happens, though, is there is a mitotic inhibition and an apoptosis of fetal cells, which is probably the background mechanism for the pathogenesis of the various defects. And we adapted the virus to human cell culture, and passage of the virus showed that it, through various markers, that it was attenuated. And this became then a candidate vaccine virus. Well, of course, there were others working on vaccine development, and those strains were grown in primary cells. In 1968, there was a large meeting at NIH at which the various vaccines were presented, and Albert Sabin was there, and he was going through the halls and saying that we should not use human diploid cells, but you know, what could I do about that? But then he, towards the end of the conference, he got up and he made this statement: there is no full characterization for any cell line, blah, blah, blah. There is always hypothetical something for which we cannot test. Therefore, the WI38 cells are no more fully characterized, blah, blah, and there's no technology, et cetera. Well, he did this in his usual rabbinical style, and but honestly, I remember very clearly, I said to myself, the Lord has delivered him into my hands. Because I got up and, among other things, said that despite my great and sincere respect for Dr. Sabin, I think the statements he has made are strictly ex cathedra and without a factual basis. So my conclusion is what we are dealing with here is theology. And you see, in theology, it is very difficult to disprove the existence of ghosts, but this is not, to my mind, a basis for making intellectual decisions. Now, this was certainly an impudent thing to say for a young man, but I received an ovation. Now, the ovation was not for me personally, but because most of the audience agreed with me. However, the FDA agreed with Sabin. And it was only something like 7 or 8 years later, after the vaccine had been used extensively in Europe, where they had a much broader point of view. It was only then, and because of the better immunological properties of the particular so-called RA273 strain, that the vaccine was adopted by Merck in the MMR vaccine. And the happy end of the story is, or at least towards the end, I hope, of the story, is that this is the map of countries now using rubella vaccine. And you can see that the only notable exceptions are sub-Saharan Africa and India, and that there is actually a reasonable argument for thinking that rubella could be eliminated from, and congenital rubella, more importantly, eliminated from the world by vaccination, particularly with together with measles. Lesson 5 is don't get into a mental rut, because having worked with diploid cells for so long, and then beginning to think of developing a varicella vaccine, we made a mistake. Now, we found, and this was the abstract that we published, we found that varicella virus could be grown in guinea pig cells. And that was sort of interesting, but we didn't think about that deeply enough. So at that point, Michiaki Takahashi in Japan was trying to develop a varicella vaccine. So was Maurice Hilleman at Merck, and also ourselves. And as you can see on this slide, both the Hilleman strain and the strain that we were working with, Webster, was serial passage in WI38. But Takahashi had grown it first in human cells, but then passage it in guinea pig cells. And he achieved what we did not. That is to say, the vaccine virus became attenuated, having been passage in the guinea pig cells, whereas the two other strains were insufficiently attenuated, even after long passage in WI38. So again, Don't fixate on things. Think about them before you try to repeat yourself. Lesson 6, I don't know whether it was Yogi Berra or Casey Stengel who said this originally, but it's not over till it's over. You all remember that in the late 90s there was a rotavirus vaccine called RotaShield, which was certainly effective. That is, it did show protection, but it also caused intussusception, and at a rate of approximately 1 in 10,000 first doses, and so it was taken off the market. It was actually offered to developing country manufacturers if they would use it on the grounds that it would prevent more deaths than it would cause, but in what might be called a profile in cowardice, none of them adopted or that offer from the original manufacturer. Well, so the need for rotavirus vaccine persisted. In the U.S., as you can see, the issue is hospitalizations rather than deaths, but many thousands of hospitalizations and medical visits, whereas the more serious problem in developing countries is something like 600,000 annual deaths and millions of medical visits, and of course, not always successful medical visits caused by the dehydration of rotavirus gastroenteritis. So, well, I'm going to skip that and talk about the development of the pentavalent vaccine. Working with two of my colleagues, Fred Clark and Paul Offit, at Wistar and Children's Hospital, and having as background the work of Ruth Bishop in Australia isolating the human rotavirus, and the work of Al Kapicki in the NIH and his colleagues showing the importance of two surface proteins, the VP4P protein, the VP7G protein, carrying neutralizing epitopes, and having at our hands the technique of reassortment, we're able to make a vaccine consisting of 5 different strains, and that is shown on this slide. What was done essentially was to go to the veterinary facility of the University of Pennsylvania and to isolate from a cow a virus called WC3, which is shown in yellow. The rotavirus has 11 double-stranded RNA genome segments, and that makes it easy to reassort. So taking 4 human viruses which have on their surface these G and P proteins, one is able in cell culture to make reassortants based on 10 segments of the bovine virus, which gives you the attenuation, and 1 segment from either a G or a P, that is segments coding for either G or P proteins, so that one ended up with 5 different viruses, each one giving a particular immune response to the common human rotavirus strains. This was then tested in 70,000 infants to show that it did not cause a significant number of intussusception reactions, and the efficacy of the vaccine proved to be very high, as shown here, particularly 98% against severe disease, an effect lasting for at least 2 seasons. Now, the effect of this in the U.S. is shown on this slide, and actually the last season, 2009-2010, shows even lower rotavirus activity in the United States. And since not every child has been vaccinated, there is definitely a herd immunity effect occurring here. And of course, we are all concerned about serotype replacement, as has happened with pneumococcus, but so far that has not happened, and this success is now being repeated in other parts of the world, and especially now we have a second rotavirus vaccine that has also been proven to be effective. A problem has arisen similar to the poliovirus problem in that the effectiveness of rotavirus vaccines in tropical countries is much diminished in comparison to its efficacy in developed countries. As you can see here in Nicaragua, the efficacy against severe diarrhea was 58% against very severe diarrhea that would probably, without treatment, have resulted in death, 77%. The reasons for this are being studied. I'm trying to push Merck into doing a trial of neonatal doses of rotavirus vaccine, and we don't yet know what the biology of this is, but it will be very important to determine that. But nevertheless, if you look at the public health effect of even this kind of efficacy, the reduction of mortality is going to be considerable. Lesson 7 is be skeptical of expert opinion. Now, you all know that literally thousands of people are trying to develop a vaccine against HIV over the last 20 years or so. So what have we learned? Essentially what we've learned is that broadly neutralizing antibodies against HIV are difficult to produce. Nevertheless, in experimental animals, you can show that antibodies do protect. So that's important. The second is that cytotoxic T cells, if present at the time of exposure, decrease viral load after a challenge. But, not surprisingly, CTL does not prevent infection, but it does have an effect on the amount of virus. The other thing that we've learned from HIV vaccinology is the importance, the interest of what are called prime boost regimens. That is to say, where you prime with a vector carrying certain genes, the HIV genes, for example, or a DNA plasmid expressing HIV genes. And this priming produces at least a feeble, measurable immune response. But when you boost with another vector or with a protein, you get an enhanced immune response. And this sort of prime boost regimen, I think, has application to other vaccines as well. At any rate, in the 1990s, we were working at Sanofi Pasteur with a vector called canary pox, which, as the word implies, is a poxvirus of canaries. And it has the property of being innocuous in humans, and also the property that vector immunity doesn't develop against it. That is to say, you can get repeated doses of canary pox and not develop a response that prevents the inserted gene in canary pox from being expressed and stimulating the immune system. So the Thai government and the U.S. Army decided that they would do a trial in Thailand in a population exposed to HIV using the poxvirus, canary pox prime, and the GP120 glycoprotein boost. Well, a who's who of HIV vaccinology, I'm sure you'll recognize some of the names on this list, published an article in Science saying this study should not be done. It's a waste of money, a waste of time, and it shouldn't be done. And you know, I respect every one of the individuals who authored this article. Nevertheless, at the end of the day, lo and behold, the trial showed an efficacy of somewhere around 30%. This is the first time that any efficacy has been shown against HIV acquisition by prior vaccination. And this despite, as at the bottom of the slide, you can see the CD8 T cell response, which the experts say is absolutely essential, was poor. So why does it work? Why did it work? We don't know the full answer to this yet, but it appears that it works through stimulating antibodies and probably non-neutralizing antibodies, antibodies that act through antibody-dependent cellular cytotoxicity, or perhaps other mechanisms. And interestingly, the antibody is directed not at the V3 loop, which is the loop that most people think is very important in inducing neutralizing antibodies, but the V2 loop, which is the loop that attaches to integrin. So in other words, it may be that this vaccine is preventing the attachment to integrin receptors on cells and therefore decreasing acquisition of HIV. But anyway, as I said, be skeptical of expert opinion. So my eighth lesson is be patient, follow your dream. In the 1970s, I started to work on cytomegalovirus. Why? Well, several reasons. One, to prevent congenital infection, which occurs in 1% of all live births and in many cases leaves sequelae behind, primarily mental retardation and deafness. Second, CMV is still the major infection complication of transplantation, both solid organ transplants, recipients who receive an organ from a seropositive donor, or seropositive bone marrow transplant patients who are immunosuppressed and reactivate their latent CMV. And then finally, there are data suggesting that CMV may play a role in atherosclerosis and, important to some of us, immunosenescence. Starting in the 70s, we developed a live attenuated strain, and I won't go through these data, but the strain produced antibodies and cellular responses, and so that was the first step. We also used the canary pox vector to show that if we inserted the major stimulator of cellular cytotoxicity, the so-called PP65 matrix protein, we could induce CTL in volunteers, shown by the black triangles, after 2 doses of the canary pox vector. More important, at this stage, in 2010, the live attenuated strain is still being studied. It has showed protection against severe transplant disease, so that's one major finding. The third item, subunit GP glycoprotein, has shown protection about a short-term protection of about 50% in women exposed to CMV. So that's another proof of concept. And the next line, DNA plasmids in transplant patients, bone marrow stem cell transplant patients, has shown diminution of viral load and reduction of the need for antiviral therapy. So we now have at least 3 proofs of concept that a CMV vaccine is possible. And we have at least 4 major companies developing a CMV vaccine. So I feel in any case that I've been successful in getting things moving, and I hope to live long enough to see a licensed CMV vaccine. Lesson 9 is stay abreast of new techniques. As a consultant in this decade, I have had to deal with a number of new techniques, and I just want to mention 3. You all know that the papillomavirus vaccine has been developed using virus-like particle technology, basically a self-assembling protein, L1 in the case of the papillomavirus vaccine. But VLPs are being applied to many other viruses. For example, influenza, where 3 proteins are required, the hemagglutinin, the neuraminidase, and the matrix 1 protein, which can be assembled into virus-like particles and constructed in vitro. But this is also being applied to Ebola virus, to chikungunya virus, and even to HIV to make immunogenic particles that are not infectious. The second new technology I would mention is adjuvants, we have learned the importance of the innate immune system in stimulating the adaptive immune system. And so we are now dealing with adjuvants that are TLR stimulators, TLR agonists. They are mentioned at the bottom. We now have one licensed TLR agonist in the SmithKline papillomavirus vaccine, but also the TLR9 oligonucleotide, so-called CpG TLR9 agonist, is proving to be interesting, for example, in hepatitis B. This is a study in the elderly, and I must tell you the elderly are people over 40 who don't respond very well to hepatitis B vaccines, to the standard vaccines, but to a vaccine called Heplisav, which contains the TLR9 agonist, they do respond. So here you see that after a single dose of the TLR9 adjuvanted vaccine, 79% of the subjects respond with antibodies, and after the second dose, 100%, compared to only 18% with the standard vaccine. Now, this is going to be applied to diabetics, to patients on hemodialysis, etc. And this, again, is proof of the idea that the adjuvants of innate immunity are going to prove to be very important in vaccinology. And the third new technology is what might be called the omics. Reverse vaccinology or genomics was developed initially by Rino Rappuoli in Italy, and essentially what it consists of is a sequence of a bacterial pathogen, sequencing the genome, selecting the open reading frames that are likely to produce proteins, growing those open reading frames in E. coli, in other words, producing the proteins, injecting the proteins into mice, and then taking serum from mice and looking for bactericidal or other functional antibodies, and then selecting the proteins that give those responses and putting them into a vaccine. So Novartis now has a pentavalent meningococcus group B experimental vaccine, which is in phase 3. You know that we don't have a group B meningococcus vaccine. It's the missing link in our protection against meningococci. And this is, well, it says phase 2 in this trial, but it's actually in phase 3, and it appears to be doing quite well. But many other bacteria are also the subject of this genomic analysis: group B strep, pneumo, anthrax, chlamydia, and even Piriformonas gingivalis, the cause of a very important worldwide disease, periodontitis. And as I said, there are other omics, the transcriptomics, that is looking for messenger RNA expressed in infection, that is in vivo, so you know it's important, the structure of proteins, what are the important host responses to give you clues as to what the protective responses are, and even looking for the genetic, let's say the genetic structure of people who react to vaccines, who have adverse reactions. So what are some of the future targets? Well, this is a mixture of, first of all, of vaccines that I think are likely to be licensed in the near future, dengue and meningo B, for example, but also future targets. For example, nosocomial bacteria, MRSA, Clostridium difficile, and we now have promising results in chronic infections. For example, women who are already infected and are showing neoplastic changes after papillomavirus infection. We know that the E6 and E7 genes of papillomaviruses are the ones that cause the oncogenic transformation, and there are already data showing that immunizing against those can actually, in a sense, reverse the changes that are produced by the viruses. And of course, many non-infectious diseases are the subject of vaccine development. So there are, however, remaining problems, and I've listed 6 of them that I think are important. The induction of long-term B-cell memory, both effector and central memory. The induction of cellular responses that are equivalent to those after natural immune responses, because when you get an infection, you mount a very significant response. We have not succeeded in duplicating that yet in our vaccine work. Overcoming poor neonatal antigen processing, because we do so poorly in the first year of life with immunization. Overcoming T-cell exhaustion in the elderly. Well, why is it that the elderly respond poorly to vaccines? It is, in fact, because their T-cells are naive T-cells are no longer being produced. Better induction of mucosal immunity with non-living organisms. And specific adjuvants that don't give you a broad response, but rather a targeted response to, let's say, dendritic cells, to T regulatory cells, Th17 cells, etc. Now, solving these problems is not necessarily going to be easy. Some of them will be very difficult, some of them will be intractable, but they will all be intellectually stimulating, which leads me to the last lesson, and that sometimes the reward is in the journey, not the arrival. And I think this is what Louis Pasteur meant when he said, young people, entrust yourselves to those sure and powerful methods of which we only know the first secrets. And whatever your career, don't let yourself fall into sterile skepticism. Don't let yourself be discouraged. Live in the serene peace of laboratories and libraries until the moment when you will have perhaps the immense happiness of thinking that you have contributed something to the progress and well-being of humanity. But whether or not your efforts are favored by life, when you approach the end, you must be able to say to yourself, I did what I could. Thank you.